I welcome you all for the post-lunch third session of the STTP. On behalf of ENTC Department of Cummins College of Engineering, Pune, I, Nitin Palan, welcome Dr. Shiladitya Mitra, Senior Postdoctorate at Max Planck Institute of Psychiatry, Dr. Prachi Mukherjee, HOD ENTC Department, Dr. Anita Patil, Coordinator of the STTP, and all the participants. It's my immense pleasure to introduce today's speaker, Dr. Shiladitya Mitra. Dr. Shiladitya Mitra is Senior Postdoctorate at Max Planck Institute of Psychiatry. He has completed his postdoctorate at European Molecular Biology Laboratory, Nankai Institute under guidance of Professor Leszek. He completed his PhD from CSIR, Center for Cellular and Molecular Biology. He has completed his BTech in Biotechnology from Vellore Institute of Technology. During his PhD, he was awarded with CSIR GATE Junior Research Fellowship, GRF, and CSIR Senior Research Fellowship. He won the Best Poster Award by Society of Neurochemistry at International Conference on Recent Advances in Molecular Mechanism of Neurological Disorder, New Delhi, and also at the Brainstorming Meeting and Workshop on Proteomics, Hyderabad. He was awarded Travel Fellowship from Wellcome Trust DBT and Travel Award from Department of Biotechnology, DBT India, for attending and presenting poster at FEBS EMBO Paris, Ambo Paris. We are truly honored to have you, sir, here. Today, he will be delivering a session on a brain-machine interface. So now I request Dr. Shiladitya to start the session. Over to you, sir.
Right. Hello. Thank you very much for this kind introduction. And I welcome everyone on this presentation that I'll be giving. And just one second, let me connect. Okay. Is this presentation being shared with everyone? Can I get a feedback?
Yes, sir.
Yeah, it's
Okay.
in here.
Right, great. So what I'll be talking today is about brain-machine interface. Like it's getting popularity for quite some time now. It's a very interesting part of neuroscience research, electronic research, or more about interdisciplinary research. But the crux that I would want to point and want to put forward during this presentation is not just giving you information about what's happening and what's exactly right now the cutting edge technology in brain machine interface, but also to share with you information about how you can do it at your home, how you can access it in your college, and that things are getting cheaper and it is no longer in the realms of science fiction, but it is becoming more or less a scientific reality. Since I am a neuroscientist, so there will be a lot of biology coming in, but don't get intimidated by that because a part of understanding the brain is the basis of how you can design a better brain-machine interface. So I'll not delve much into biology, but at the same time, I'll give you enough so that you have the idea of how the brain-machine interface works, and you can troubleshoot and design, in fact, on your own. So everybody knows that this is high school stuff, that the body has a lot of electrical activities which are going through. You have the brain, you have the heart, you have the muscles, and they communicate and function through electricity. These electrical activities can be measured, and particularly if it is for the brain, then it is called an electroencephalogram, which is an EEG. If it is for the heart, everyone knows it's called an ECG, an electrocardiogram. And for muscles, it is called an EMG or electromyogram. So what it does is that you can put on receivers that amplifies the signals that's happening and it gets transmitted. You deconvolute the signals or you read it as such in cases of like electrocardiogram, and then you decipher accordingly the interpretation of the data that is coming out through. So these electrical impulses that are getting created in our body, in the brain, in the heart, in the muscles, they are actually created through the exchange of ions, which surrounds the cell and the surroundings and which are inside the cell. So particularly, like, I'll go for the neuron. So this is like a basic model of a neuron. It has, don't bother much, it has, let's say these are the receiver parts of the neurons, and this is the transmitter part of the neuron. It's called the axon, and these are the dendrites. And all throughout the length of the dendrites and axons, you have multiple channels, like gated channels, which open and which close. So and allow ions to flow in or flow out. So it depends upon what concentration of ions are flowing in versus what kinds of ions are going out, is that it changes the potential across this axon. And when you have a change in the potential across the axons, then you generate an electric current. And this is how electric currents propagate from one neuron to another. And the mechanism is the same when you are considering a muscle, which is a heart muscle, and that's why you get an electrocardiogram, or if you have a muscle in your arms, which you can contract and relax. So the neuronal model, and because of the change in the ions, you can get an action potential. This is called an action potential. This is also the firing of a neuron. So you can have failed initiation, or you can get a spike like this. I was saying that since the neuron is so electric, there have been people who have been trying to model a neuron based on the electrical components that we have. So this is an example of that. So this is a 3-transistor based modeling of a neuron, where you get a stimulus and then you get an output through an oscilloscope. And as you can see over here in the oscilloscope, you get this fast spiking nature. So in fact, in nature, there are fast spiking neurons which are already there. Of course, this is just one example. There can be like different models of neurons that are available. And if you want, I have the link at the end of this presentation, so you can use that software to design your own neurons, and those are the basis of neural computing. So I'm moving to the next slide. Please give me feedback that it moved. So I was talking about that, like I was telling about neurons, single neurons in my previous slide. But the neurons in the brain are much more compacted. So they are arranged in an array like this over here. In the first image on the left-hand side corner, as you can see, this is an image of a mouse hippocampus, which is just one region of the brain involved in memory and learning. And you can see how densely they're arranged. So as you know, I was showing you an electrical the action potential of a single neuron in my previous slide. But when their neurons are so densely arranged and they're communicating with each other, it's like electrical activities which is happening inside your room. You have different devices and they all have 60 hertz, as you know, that or the refresh rate is somewhere between 50 or 60 hertz, or you can have even 120 hertz cell phone screen these days, but they have this background noise. So even in the brain, there are some background waves or background noise. I wouldn't call it noise, but background waves. And these background waves are the ones which are depicted in this image over here. There are, can you see my cursor moving over this image right now?
Yes, yes.
Yeah, okay, great. So there is gamma waves, there's beta, alpha, theta, and so on, so on. And each of these waves are basically the ways of how the neurons are communicating with each other, how the background activity, electrical activity is happening in the brain, and each tell or indicate towards a specific state of the brain. So like if you are seeing this fast frequency, like compact frequency, very high frequency waves over here, which is called gamma waves. So that usually happens when you are trying to concentrate and when you're trying to do a problem solving approach. Beta is more about a regular alert mind, like when you're alert, when you're paying attention, listening to a lecture or something like that. And alpha is more in a meditative state, like when you're relaxed, you're sitting by having a tea or something. Theta is more about drowsiness, or even it can be about creativity. When you are in your daydreaming state, when you're half sleepy, you are trying to understand. And the delta can be like really long waves, which is mostly in a long REM sleep. So these waves are actually the basis of how most of the brain-machine interface devices interpret the brain and are designed. Am I still audible?
Yes, sir.
Oh, great. Finally, the connection is not getting broken. Great. I'm happy about it. So yes, so these are the basis of how the brain-machine interface devices work. So they usually will be picking up, so suppose most of the devices which are available in the market, which are available off the counter, and like the simple ones will be picking up either beta, alpha, or the gamma waves, not much of the theta and the delta over here. So what you will be doing with these is to use simple attention based, which I'll be coming and explaining later on, simple attention based jobs that you can assign. And once you get this, you can just deconvolute these data and put it through another software, which I'll also explain in the later. But so these brain waves can be found through but remember that these are recorded from the cortical regions of the brain. These are not deep brain waves, and they propagate throughout the perineural system of the central nervous system. So to understand how a BMI works, first you have to understand that how do you measure a brain's electrical activity. So since the brain is electric, so it has an electrical component and it has a magnetic component to measure. So when I'm talking about the electrical component, first I'll be talking about a non-invasive method, which is the most popular one, which everybody has seen. And I shall stop here again to ask if I'm still audible.
Yes, sir. You are audible.
And are you okay? And are you great? And are you seeing the slide right now, which says that how to measure brain's electrical activity?
Yes. Yes.
Great, fine, fantastic. I'm so sorry about the delays before. Okay, so let's pick it up. And so you have the EEG, which is an electroencephalogram over here, and this is how it basically looks on a person's head. You have different electrodes which are placed on a person's head. But they are not just random electrodes which are placed, they are basically placed based on a brain map, which has been coming down and handed over to the informations of science. So when you see the nodes in which the electrodes are placed, these are diagrammatically represented over here. And the most common nodes that you will be finding is FP1, FPZ, and FP2. These are the prefrontal nodes. And this prefrontal cortex is the one which is involved in active decision making, in attention, and in most of the decision making tasks. You also have the frontal cortex over here, these marked, which is with this green, and then you have the parietal cortex, which is mostly with associations. So depending upon what action you are doing, each of the electrodes will be firing differently. And then, of course, this is a very dense array. You can have as dense an array of electrodes as you can go to 30 to 16, but then usually it is 10 to 20, and 10 to 20 is enough to give you information about the function of the brain and each of these brain regions and how they are functioning. So usually what happens is that you associate these electrodes, and then an amplifier records the signal from the brain, particularly from the brain's cortical regions, not from the deep regions. Again, they use Fourier transform or some other transformation paradigms to transform the signals that are coming from the brain, because you'll be getting a mix of signals. So you have to deconvolute it into the specific signals and the specific frequencies that you want to see. Like it can be a mixture of gamma and beta, but you want to deconvolute it, or it can be just background noise. So you do that, and you can do that using MATLAB, Python programming, which of course you guys know. If you don't know MATLAB and Python, that's also fine, because nowadays there are multiple softwares over there, which I'll also be talking about, which can decipher the EEG data for you. So these have some specific codes which are written, but then those codes are not so great. Like it won't give you an accurate data, but it'll work. So far, so I'm sorry, I think I exited the presentation mode. Yeah, and it gives you a functional and structural correlation in tasks and interacts with the environment. So whenever you do something, each of these electrodes will be firing up, as I told you. Am I still there?
Yeah, sir. Yeah.
Right, okay, great. So I'll move on to the next slide. And these are, so that was a non-invasive technique. And this is an invasive technique. So of the electrical component. Like the non-invasive technique, this also measures the EEG and is called an electrocorticography, or flexible brain implants, which is microelectrode array. There's a little bit difference between these two. While electrocardiography is more like an invasive EEG, where you place this array, you open the skull of the brain person, and you place this array directly onto the brain, and then it collects local field potentials. And this is called an invasive EEG, and you get much more information out of here because you don't have interference of the skull. But and when you compare that to the microelectrode array, they are like electrodes like sorry again. There are electrodes like here, which as shown, they have got really dense fibers, which are microwires array, which are running down. And you put this into the brain in one specific region, based suppose somatosensory cortex or primary motor cortex. So these regions are involved in understanding the senses that you're getting from the environment and for your movement. And these pick up data from many neurons now. And when you pick up the data from single neuron firings of multiple neurons, and the data looks something like this. And these blue regions that you are seeing are actually firing over here, or a synchronization of firing, particularly when the subject was interacting with the environment. So you see a pattern like this, but like it's not like a continuous. There are like multiple small lines over here that says about each of the neurons firing over here, and so you get like a picture like this. And then you decipher the picture based on the algorithm that you put in and how it is interacting with the environment and how the neurons are synchronizing. So the good thing about these invasive method of measuring the brain, particularly like the brain implants, are that they can act as both input and output, unlike EEG, which is a non-invasive and just outside. So you can record the data from the neurons, and as well as send electrical impulses through to mimic certain stimulus that these neurons are getting, or to fire them, which are also an important part in a brain-machine interface, which we'll be seeing as few examples later on. And again, apart from the electrical component, there is the magnetic component. And for the magnetic component, you have the magnetoencephalogram, which is similar to that of the EEG, except the thing is that it is much more precise. It measures the neuronal activity directly instead of EEG giving brain waves, and not like an overall activity measuring, and not like specific. And then you have the MRI. Everyone knows the MRI. These are the magnets, a strong magnet that allows the flip of the hydrogen, and then when it shifts back to T1 to T2 phase, then it collects the information, and that's how you generate a structural map or a functional map of the body and the brain. So, but then these two can be brought in together to call something called a magnetic source imaging, where you have the information of very small voxels and the information at a single neuronal activity level, and that can generate a beautiful map like this. This is not an artwork. This is, of course, like it's rendered, but this is based on data that you will be getting from magnetic source imaging with a very high quality MRI and MEG, showing you the connections in the brain, different brain regions, and how they're connected to each other. The biggest drawback, however, of the magnetic encephalogram and the magnetic resonance imaging as compared to EEG is that it is prone to interference, they are costlier, and they are extremely sensitive to like external changes in the environment. So they are not so portable as EEGs are. EEGs, that's why, are much more used in everyday research as compared to MRI. So these, I'm still there, right?
Yes, sir. Yes.
Right, great. So I'm sorry. After a few slides, I'll be keeping on saying, because after
Don't worry, sir. I'll inform you.
Right. Okay. Great. Okay. So, like those techniques that we have been seeing before, these techniques are used to understand how the brain works, how the subgroup of neurons works, and how they're connected to each other. And they are important because because the brain is a complex organ. It is not that very simple that one organ works uniformly. each of the sub-regions of the brain, as I showed in the pictures previously, has a different function. Prefrontal cortex, somatosensory cortex, the hippocampus, the amygdala, they have different functions. And along with fMRI and the PET, people and the scientists around the world are trying to understand how each of the regions connect to each other, how they are working. Historically, in fact, clinical studies for patients who come with disorders that have been giving us knowledge about how each regions are functioning. And that's also the basis of very early EEGs in the 1950s, where you didn't have so many high quality technologies. And again, we have preclinical models like in rodents and monkeys, I mean like mice, rats, and monkeys, in which you use even more interesting and cutting edge technologies to understand the function of neuron ensembles and the brain plasticity, which includes optogenetics, in vivo multi-trode recordings, calcium imaging, immunostaining to understand the functional aspect of the brain's neural connections, of which I'll be talking about one because which has the potential of being introduced in BMI, but that will come later on. So I'll start now, since I have established the background of how you get the data from the brain, how EEGs work in principle. It's actually now interaction with the technology. Now that you have the data, now you have the full access to how you will be analyzing the data and what you'll be attributing to it. So let's start with interfacing with technology. The first I'll be talking about the non-invasive ones. Before I go into the typical EEG stuff that everyone knows, let me share with you something that is that is to be considered when you're designing a BMI, particularly a very good BMI. So this is an experiment which was done by Libet quite a long back, actually, in the 1950s. And this was an experiment to show that the brain, your subconscious, actually decides before you decide something consciously. So it asked a lot of question on free will, but the idea is that we are not going to the free will debate over here. For the reason for me to show you this data over here is because to show that brain activity is much more faster than conscious actions. So when in EEG you'll be picking up a brain activity, it might be before you are actually doing a task. And this will be important to when you're designing a BMI, because the brain activity might be not in synchronization to your conscious decision, and then the person with whom the BMI is there can not be in sync and can feel it confused. So what this experiment did was that they gave the person who were in the experiment a choice to do a conscious decision, and then they were making analysis of the EEG, and they found out that 200 milliseconds before the person thought the person made a conscious decision, the brain had enough evidence that the decision is going to be made. And this was a pretty old study, and very recently, not very recently, it's still been 12 years, there was another study which was done with MRI and with much more stronger technology that showed that this decision making can be even as large as 10 seconds, not just milliseconds, but 10 seconds before a person can be aware of a decision that's being made and the brain is encoding the decision to be made. So this can, with this, you can design a very simple brain-machine interface cheating game in which you can have a button which has a delay of, say, suppose n seconds before it turns red. So you tell the subject to press the button before it lights up or turns red. And initially the subject is very happy. It presses the button at will before it lights up. Some are misses, some are hits, but that's the entire thing. And the subject is really happy. And then you record the subject's EEG and you feed the data of the person's brain over here. And then the EEG can determine an activity like this. And when you do put this for a machine learning, and you can pick up the signals before actually the subject is doing an action. So then what happens is, because this device is now connected to a feedback to these lights, what you can do is that before the person picks up his or her hand to press the button, the lights go on. So now you have a cheating device over here. So that person will never, or at max, in a very statistically less frequently, will be able to press the button before it lights up. So this is the basis of a lot of experiments that happens, particularly, like you have the movement of big prosthetic arms or something, but these are really cool experiments that has been done. And what this information tells us, this is the picture from the same experiment of 2008 that I was talking about, is that this is the prefrontal cortex over here, and the prefrontal cortex and the parietal cortex over here fires up. So these are activity patterns that you are seeing, which gets increased when the person is doing a conscious decision making over here. So as I was telling, that why this LF1, LFZ, and LF2 regions of the encephalogram are important in designing a BMI, because most of your signals of decision making or attention comes from the prefrontal cortex over here. So talking about this, like now that like this is the brain activity is really fast. So let's talking about a basic design of a BMI, which is the basis of every BMI, like whether they are really scientifically, really science fiction ones, or which you can do it by yourself. So you record the EEG while the person is performing a task. It can be modular, or it can be a very, for easy analysis. It doesn't have to be very complex task. So you can attribute each thing, like, so suppose tell that person, look left, look right, and then you see the firing patterns over in each of these electrodes which are placed on the brain. So this is the map of the electrodes which are placed on the brain. Each of them picks up different attributes. Like the F7 picks up verbal expression. So when you're going to talk, it causes the muscles to flex, and it picks up that as well. These are the prefrontal one, which picks up for the attention, and so on and so forth. So then you put this data that you're getting. If you're a good programmer, you use MATLAB and Python and apply a machine learning approach to this. If you are not such a good programmer, there are, as I told, different softwares which are available, and you put it through there. So when you put it through there, like you get, when you put EEGs before machine learning for data analysis, you get a signal something like this. These are each of these different electrodes which are placed over here. You get activity like this, and you get a map like this, and this map keeps on changing. And these activities, as you can see, that there are a lot of ways, but they are not deconvoluted, and the deconvolution happens when you are applying the machine learning. And then once you have deconvoluted, and once you have and once you have set us, like trained the brain to understand a certain, say suppose left or right, then you define an output task. And then when you have defined an output task, you can upload to a microprocessor or a microcontroller to execute this task, and thus designs the instrument part or the machine part and you can interact with the environment with this. I'll first come to one of the most popular examples when you are talking about brain-machine interface is this was in FIFA World Cup, which happened in Brazil, and the World Cup was actually kicked. The first ball of the World Cup was actually kicked by this person who is a paraplegic. So that means that he has a spinal cord injury, and he has, after the fourth vertebra of the spinal cord, he does not have any sensations below. And this was for a very long time, like 5-6 years, he did not have sensation. But this is a prosthetic exoskeleton that the person is controlling with the person's brain and is allowing him to move his leg. I'll detail how this exoskeleton functions. So it's not very sci-fi. It's again very easy to design. The exoskeleton function over here, and the person hits the ball, and of course you can see the enjoyment and the joy that person who has never been able to walk for the past 6-7 years in hitting the first ball of the FIFA World Cup. So this is how it actually happens in the lab. Like you have this device, the person is attached to wires, helps him to move. The EEG, and this is a non-invasive one, so you have the EEG connected on the person's brain, giving in signal to an array of computers over here, and each has hydraulic. So over here, the exoskeleton has hydraulic motion, so that like the motion is dampened, it's not so fast. And if you train a brain about how to move, this movement, and then it teaches that person to move the leg, or the exoskeleton makes the person move the leg. But there are some interesting things. When the person moves the leg, the person also gets a haptic feedback in the person and the arm, where he can still get the senses. So when you have the haptic feedback in the arm, the brain is such a beautiful organ. And if you get time, you can read about Ramachandran Guha's The Phantoms in the Brain, that there are connections which are still existing, even though that person is paralyzed from the fourth vertebra down. And the brain attributes that this haptic feedback to the action performed, and he can actually feel. That's the reason his first statement when he hit the ball in the FIFA World Cup was that I felt kicking, though he can't feel kicking because he was completely paralyzed. So there are some very interesting things when you design a prosthetic or an exoskeleton. One of the most important stuffs, apart from the designing component, which is like any BMIs, when you give the feedback from the brain to the and train the arms first virtually using a virtual algorithm, and then you make the person arm move, is that you do it with a positive reinforcement. You let the person walk in front of a mirror or in front of a computer diagram. And these actually sometimes fire some specific neurons, which are called the mirror neurons, that in case of this person, which doesn't have any senses, does not, maybe these neurons from his spinal cord are completely not working, so he does not have the feeling of what is to move a leg. But then these can mimic those feelings and thus start, in fact, help rebuilding it. So and also another very important thing is that you need to get haptic feedback. So as I told, that he gets haptic feedback through an EMG in the arm. And before doing this, he was trying to get different textures, and with each textures, he was getting a different electrical impulse in his arm. So he could relate to each of these haptic feedbacks as to different textures. And the brain is an amazing organ. It starts picking up and translating these haptic feedbacks that you get into different situations. So these altogether helps you design a really interactive exoskeleton, in which you not only just program the movement of the limbs by the exoskeleton, but also get the haptic feedback to settle that, like the person and who is doing this. In fact, the beauty of this is that there were 8 people. Am I still there?
Yes, sir. Yes, sir.
Right. Okay. So the beauty of the system is that there were totally in this. So the scientist who was performing this is Nicolelis. He is one of the pioneers in brain-machine interface, particularly when it comes to the non-invasive ones. You can go up and look at his papers. They had done a lot of experiment on monkeys initially before he decided to do this over in human beings. The interesting fact is that in doing this experiment, he's a Brazilian actually. That's the reason he wanted a Brazilian to kick the FIFA World Cup football. So there were 8 subjects who were involved in the experiment. And of course, since 8 subjects were involved in this experiment, so they have to be also put for a long-term study. And when these 8 subjects were placed for a long-term study, they found that 6 out of these 8 subjects had been showing actual recovery. Like they could show signs of movement even without the prosthetic limbs, even without the exoskeleton. And these are actually something where BMI is coming into action in therapeutics, that it is not just helping you to move, but it also helps you to recover, which is a spectacular thing, because no doctors would be saying that that first recovery could happen. So, like this is the overall diagram of how. Yes? Hello?
Yeah, participants, please switch off your mics. Dr. Morankar, please switch off your mic. Thank you. Yes.
Okay, so yeah, sorry, I'll continue. So yes, so these are the different ways, like the neuro rehabilitation training goes on. So you do the EEGs, you still give the haptic feedback, which is again really crucial for the brain to think that. So this is again an example of brain over body. Like once the brain starts to think that like there is a positive reinforcement, it starts to recover the spinal cord. And then when you do a 0G test, in which you hang the person from the like over here, you hang the person from these wires over here, so that the person is not actually putting his entire weight on the ground. So it's shown that the person starts slowly moving his legs or her legs. And after some time, you can do this and make that person really, like after many times, it can be more self-sufficient. It takes a lot of time, but over the course of a year, a lot of patients were able to. So this is something, is BMI is not only just for playing games and just for designing the neuroprosthetic, which is actually great for people who are paralyzed like this person, which I'm giving as an example, but also it can help to rehabilitate the person and get that person better medically, which is, I think, a great potential of brain-machine interface, particularly for a psychiatric researcher like me. But again, like these are in the realms of still of high quality research, and very expensive stuff. You really need a lot of people. There were altogether 200 people who were working for this project of to make this person kick the football. So understand there's an entire team working over there. So, but then this should not dissuade you from thinking that this is something that is unreachable. In fact, that BMI has gathered sorry, so much of popularity that there are right now many affordable devices that will help you to do brain-machine interface task or design a brain-machine interface task, even if you're not a very serious programmer, and do it yourself. Do you guys know about this? Like, let me get a feedback. Like, do you guys have heard about these portable BMIs or portable?
No.
Right.
No,
Okay.
sir.
Right. Okay. So then I will also explain like these and then some of the chips. And okay. So there are multiple such companies which are there, and I'll be talking about just three because you guys can discuss, debate, play around with these things. Design robotics, which is like super awesome. So there is NeuroSky, there is Emotive, and there is OpenBCI. First, let me start with NeuroSky. They have a headset which is like this, but then the problem with this, it is only a one-channel recording with one reference channel over here. So this recording happens from the prefrontal cortex, as I told, which is very important for most of the decision making, gives you the attention. And they have a great biosensing chip, which does great amplification and decoding of the signal. However, they are done through a proprietary software. So this software tells that you are having high retention or like drowsiness, and that can help you design directly a brain-machine interface just based on attention, like moving a ball through an electrical grid, or so such kind of stuff, and such games exist. Like it's not so difficult, and it helps also to interact with the virtual environment and train basic commands. Some of this software, particularly even though it's a proprietary software, it is also compatible to open source softwares like OpenVibe, which I'll tell you again like later on, and can be given information to a microcontroller like an Arduino, and thus you can design a BMI, and it does not require real programming skills. They are again pretty easy. Each of these headsets costs something around $100 or 7,000 rupees. But it has severe limitations because it does not give you an array of information just based on the prefrontal. So the scope of things that you can do with it are limited. You can design a very simple game, of course, but if you are thinking of designing some serious robotics, then no, this will not be the choice for you. So then it comes to the next one, which is called from the company called Emotive. And they have different arrays, but what I'm going to present is the 5-channel ones and the 14-channel one. The 5-channel is around $300, a little bit expensive again from the previous one, and the 14-channel is definitely expensive. It goes around something around $850, but they're still super cheap as compared to the medical grade EEGs that you get, because they run up to, and along with the software, they can run up to tens and thousands of dollars. They give the software for free, The software is super easy and it's really good software. Based on the movements that you do, you can train the software, whether you're pushing, pulling, or dropping, and or like you can attribute them to any 4 different. You can left, right, up, down. And this software is the SDK, however, is paid. So it gets really costly from there if you want the full set of data. It does not give the raw output, raw data by default. So you cannot play around with the raw data of the EEG that you're getting. But these basics, but if you're just interested in having some basic sets of commands, the 4 commands are good enough to start with making a robot. And they do not have, because you're not getting the raw data, so you cannot open it to open source softwares like Open Vibe over here, but you can get it to link to softwares like Node-RED, which is designed mostly by IBM, and it's basically helps you connect to third-party hardwares like Raspberry Pi or Arduino to develop your BMI. And it's pretty easy, straightforward. It's just drop-in features. I'll talk about it later on in brief. And you can design your brain-machine interface device in no time. You can design a robot like a small walking, small car, which can be going left, right, depending upon the command that you're getting from the brain. And I just wanted to ask you again one more thing. Do you guys know about the Arduino? Like you must like.
Yes.
Right. Yes. Okay. So you know the Arduino, like how it works. So you can just command and program over there. Then I'm going to talk about the last one of this slot, which is the OpenBCI, which by the name says it's an open source brain computer interface. And that's their idea is. It is definitely more expensive than the previous 2. But again, since they don't have any proprietary software, or they do have like their own software, but it's not like locked in. They also is based like the same as like a Arduino design. Their chip is also based on an open platform. So like they have the chip design there. You can just go to BCI, Open BCI. You can download the chip design. You don't have to buy from them their chips. You can place an order and you can make the chip on your own. So you have the most popular one is called the Cyton board, which has like an 8 input channels, which is, I think, quite fair enough for doing any sort of small proper brain-machine interface devices. The good thing is that they're modular, so you can design the devices according to own. So like you get a headset separately, you put the headset onto getting the information to over here, and then you always get really high quality EEG data. And since they are compatible with Open Vibe, you can program Raspberry Pi or Arduino. Over here, I'll just go out and like try to show you OpenBCI's website, where like this is their OpenBCI's. Can you see the screen right now?
Yes, sir.
Yeah. Okay. So this is an OpenBCI's. If you go to OpenBCI's site, you will be seeing the screen, and this is how exactly their software works. Is that these are the different electrodes and the signals that you're getting from these electrodes. You get an amplitude, and you also get a head plot. So what you're seeing from this head plot is that when the person is interacting with the environment or doing a task, is where the signals are mostly coming from. And once you have this information, this software has the capability to assign roles, or you can just link it up to OpenBCI and assign roles to each of the functions and interpret and do a machine learning in the software itself and the algorithm. And then just convert this directly using the drag and drop feature of and put it into your Arduino or Raspberry Pi, whatever you want. You want a microprocessor or a microcontroller. So a microcontroller is more better for designing a BMI. So going back to the presentation again. So I don't have, so these are the 3 most important things which are accessory to these kits that I've been telling about. And so you have the Arduino, which every one of you knows. It's you can design the software. You can write the code on Arduino. It's very easy. Everyone does it. And then is Open Vibe, which is the software I was talking about. It's also written in C++. So it's written in a very basic language, but it's not just code-based. You can have a code-based system if you want. But if there is a GUI-based system, as you can see over here with the block diagram, you can just drag and drop the features over here. You can acquire real-time signals from, particularly for OpenBCI, you can get really high-quality signals, and that can be used to analyze. But remember, these are not for medical diagnosis. They are only for, because they are not so great, but then they're very good enough to design BMIs. And then you can like filter, acquire, classify, and for designing, testing, and using the BCI. It's also an open source, so you can just feel free to go through it, and it's super easy. If you're not a programmer, you can still do it. If you're a programmer, it's great. You can link it to MATLAB and even get really better output from it. And then it comes the Node-RED, and the Node-RED is again a drag and drop feature. You can do some programming over here also, like Emotive was connected to Node-RED, but you can design Node-RED designs like this, which follows a JavaScript program, and you can make Arduinos work with this. So the basic idea again is the same. You get the signals, you put it through these softwares, and then you get the signal, you put it through first through a biosensing chip, and then you put it through the software, which is an interface between you and the machine or the computer. Or you can, if you want to design a game, you can just design it directly. Particularly Node-RED is great for designing games because you can just put virtual commands over here. It can send you a tweet when you're thinking about a tweet. It can like look up Facebook, post for you on Facebook. You can just play a random game about it. So OpenVibe and Node-RED are great for virtual integrations. Not just brain-machine interface, but you can do virtual integrations with them as well. And here is the basic design. And if you have a classroom, or if you're a novice who wants to do it for themselves, everything can be done because they're open source and free. It can be done in less than, let's say, 30,000 rupees, which, like, of course, is expensive. But to think of the potential that it has is really fantastic. Since you are electrical, like people who are from pure engineering discipline, the real limitation of having a cheap BMI is the biosensing board. So if you guys can come up with a cheaper biosensing board, then that is great, because that is the most limiting factor apart from the taking of the data, because the EEG data, you can just pick it up from simple probes, and those probes does not cost more than 1,000 or 2,000 rupees for the entire brain. So that's not the limitation. The limitation is the biosensing chip, in which you'll get the signal and you'll filter the signal to make it into a more readable format to put it into the software. So now that I've told about the non-invasive BMI, now let's come to the next part, which is a little bit more science fiction-ish. But it's still in like this part, because it's invasive. It does not get as much as permissions as it gets for designing a non-invasive BMI, and thus is out of reach for most of the people until unless like medically prescribed, or it's still in the experimental phase. But the idea is the same. As I was telling you earlier, that you can record brain activities using EEGs, or you can directly implant the chips onto the brain. So here is the same thing. Here you implant the chips onto the brain, and it picks up the signal while the person trains to do something: read, write, move left, move right. And when you get this signal from the brains, it's like this, like you have the firing potential, and then you can get the read of this data through MATLAB, Python. Here you would require definitely a higher amount of analysis as compared to the simpler BMIs, and so the open source ones will not work. You really need to work it out through MATLAB, Python coding programs. And then once you have assigned the function, then you can move a prosthetic arm. And again, a visual and a tactile feedback is important to reinforce this process, because it helps the brain cement the connection with the prosthetics, so that the person can actually feel, the brain can actually feel that that is happening. The great part of this is that it does not only read but write. So not only giving tactile feedback or haptic feedback to the arms, it can also place these impulses onto the brain region. Say suppose a brain region that feels touch, and that person can actually feel it. At this time, it will not be like a phantom feeling, but it will be an actual feeling. So this is again a case study that I'm going to talk about. The paper came out in 2019, though this has been going on for the past 4 or 5 years now. So this is the guy who is completely paralyzed, and why I'm showing you this case study and not showing you another exoskeleton, because exoskeletons are getting pretty common. A lot of people are doing experiments with exoskeleton. I showed you one example of an exoskeleton using a chip. But this experiment, not an experiment because of subject, but this study is important because that person is able to hold things, to do work with his own arm, which is paralyzed. It's not an exoskeleton, but his own arm. And you can see that there is this window on his skull. So this is where through this chip is the chip is implanted already in his head. It's a permanent implant, and you connect the output when that person is sitting over here. And right now it has been a wireless output, so you can just connect the wireless output over here, and you can get the information into the computer that gives again feedback. So what kind of feedback does it give is that when you teach this person first through mirror neuron about the movement of his hand, so that the person can synchronize to that virtually, and then you have this device on his hand which has got lots of sensors and lots of electrodes on here, which gives small currents, EMG. This is EMG that I was talking about in the first slide. And these EMGs has the potential, depending upon where exactly it is placed, to flex your hand muscles. So if you have an EMG over here, you can move your thumb. If you have an EMG somewhere, then you can move your wrist, and depending upon where the stimulus has been given, you clench your fist, you move your thumb. So this tells how electric and actually cyborg our body is. You can place electrical signals and you can move, and this is a very easy experiment that has been given. You can put electrical signals onto your hands, and if you get electrical signals, you lose the control of your hand. It will just fall off, but at the same time it can be programmed the other ways in making that person hold something. So this is again the design of it. So you have the neural implants over here, which gives you the neural activity, which goes through, first you have to train that person to move the arm virtually, and that person is seeing the movement of the arm and thinking that he is moving it. This activates possibly the mirror neurons in the brain and thus ignites this circuitry. And then, like once this this BCI, this BCI is done, then you go for the actual prosthetic. So you have this functional electrical stimulus that it thinks that he is moving. Then you get a stimulus in the upper arm where he is not yet paralyzed, so he feels that the signal is happening over there, and then it gives this EMG signals into his arm, which actually moves his fingers, his wrist, his palm over here, and everything also gets like this is the ground wire over here, and you get a haptic feedback and a tactile feedback over here. This completes the process of a proper BMI, which is something that we expect to see in upcoming years for people who have got really serious paralysis or other problems. This is just an example. You can use this for a person who is in a completely vegetative state and not able to move their head. So can use the brain activities of that person to actually move that person. It's not only just semi-move, but though the research is still preliminary, but they are still going on in recent times. So I'll just tell you about one future directions, which I was telling, like preclinical models of rodents and monkeys are important because all of these experiments that I told you about on human beings were first performed on monkeys and first performed on rats and mice. They are, no doubt, they're called the lab rats, and then they were translated to human beings. So recently we have a very interesting technique which just came out in prominence in 2014 by Carl Desseroth from Stanford. And this technique actually utilizes the principles of the firing of a neuron. As I told, that neurons have these ions going in and going out, and that creates the electrical potential. So the idea was that if you make ions, you make the neuron to uptake ions or giveaway ions, then you can make the neuron fire, or you can make the neuron stop firing, which makes logic. So what this guy did is that he took away a bacterial design and a molecule which is called an opsin, a channel rhodopsin, which is light sensitive. So when you shine a light on this channel, it opens up and it allows the ions to flow inside the neurons, and thus creating an electrical impulse. So when you shine a blue light onto this neuron, the neuron fires. The same way it can be having another kind of opsin, which is a halorodopsin, which stops the ion from going in, thus stopping the neuron from firing, just closing the ions over here. How this is done? These are basically some proteins, and how you express the protein. So you use the, like right now you are having SARS, or you're having COVID situation. You know about viruses. Viruses go infect every cells of your body. They express inside. There are proteins inside the cells of your body. So this is done the same way, but not with such infective virus. So there are some category of viruses which is called AAV, which are non-infective, but you can which are actually still used in clinical trials in some other genetically modifications, or like gene therapy trials. So with those you can like express this protein and it does it. So you see over here, this is mice, and this has a fiber optic implanted in the cranium, and it's shining the blue light, just propagating like some kind of signal to the mice over there. And the why this technology is so great, because unlike that of EEG, you can actually reach deeper brain regions. As I told, that brain is a complex organ, so you can actually stimulate regions of the brain which is involved in tasting, in fear, in different sensory, behavioral, locomotor changes, and they have got immense potential in not only medical field but also in other disciplines of designing a BMI, or a cyborg, or a person who is brain dead, or a person who is not able to feel something, like, or a person who has gone through traumatic injury. And these are really crucial experiments. So this is one of the experiments that that I had been doing. So as I was telling you, have forget about it. So what you can see over here is an active population of neurons. So these neurons are active; they're firing, and this is the condition when the mice is just moving around, it's not doing anything else. And this is the condition when the mice is giving a choice of water and sugar. Mice love sugar; it just goes on and having sugar. So when you have a choice of water and sugar, you see the neuron firing has increased over here, and this is one region of the brain, the central amygdala. So now that you know that these neurons might be involved in giving the mice going towards the sugar water, what you do is that you express this channel rhodopsin over here. And this is a Skinner box experiment in which I'll not go through the behavioral details of it, but what it means that when you put the lace, but what I expressed over here is a halorodopsin, which stops the neurons from firing. So when you do that, you see over here that the mice stops going over to getting the food from the dispenser by itself, or getting the sugar water. So this is itself great. This is again like with fear learning and fear memory, but these neurons are associated with that. So a person who is having a traumatic injury or something, you can like give this person a different, but like this is still in basic research. It's still not gone into, it's there in monkeys, but it like going into humans will take decades, because it involves a lot of ethical issues over here. But if this actually comes down sometimes in future to human beings, we will be most probably living in a matrix-like environment where you can actually not only feel, hear the artificially created environment, a virtual reality, but also be able to sense and savor the environment, like taste it. Like the person says that the hamburger tastes amazing, or the steak tastes amazing if you watch the movie Matrix. So they are not anymore a science fiction. They are happening, a reality over here right now. So this is now, so now you know that like BMIs are been there. BMIs are pretty affordable. BMIs are happening. A lot of research is already happening. So that's the reason when Elon Musk presented about his Neuralink, not many people were impressed, including me, because the technology is not so great that he told. But what he told about is a robot that can do the chip implantation for you. which is like, okay, sure, it's not so great. But I feel that the real power of developing BMIs rests with countries like India over there, where we have got immense power over software and immense power over data analysis. And I feel that if it gets affordable, there is huge scope of this going forward, and it will be just soon when people, like, instead of virtual reality headsets, are having BMIs, or flying a drone with their BMIs, or moving a car with their, and it's happening. We are in the happening state right now. So in conclusion, I would say that, like, there are ethical concerns, of course, like, people saying that there can be mind hacking of happening, but right now we are not in the technological advancement that there will be mind hacks. We are just in the technology advancement of having jobs done with our mind. So BMI has immense application, like in games. Again, it's very simple to design a BMI in Internet of Things, in appliances, and this can be done even right now. Like, the BMI is getting cheaper, it doesn't require much even programming language. So anyone can design a BMI right now. And of course, it is involved in scientific studies, medical science, and psychiatry. Particularly right now in our hospital, we have in the psychiatric ward, for many people who are coming from traumatic injuries, a BMI that says a simple yes or no answers, but it's still great, because for people who are, like, paralyzed and or coming from like war places, to giving something yes or no is something fantastic. And I feel that the recent advancement of like the reconstructing images of a person can lead to better and interesting BMIs. So I feel that should be the end of my talk. Here are a few resources. As I was telling, that you can design electrical neurons. So if you want to go, there's a neuron software over here, and there's a book in which you can read about how bioelectric and biomagnetism field happens, which is the basis of a BMI. Right. So I hope I'm still audible.
Yes, sir, of course.
Right. Okay. Right. Okay. So that was the end of my presentation. So if you have any questions, you can ask me.
Yes, participants, any queries?
Yeah, I got a message
There
to
is one
ask
question.
for the link.
Sir, can you specify the name?
Specify the, okay. I'm getting a few questions. Yes. Specify the name. Okay, I have a few questions that says that Pranav has asked that you can specify the tool which converts the ML programming in C++ for deployment in the embedded device system. And could you paste the link here? Yes, of course. Like, you can go for Open Vibe, which is an open source software which can convert. It's designed in C++, so you can get the signal and you can do it. So I placed it over here, and for the neuron software, I'll just copy the link once and I'll just paste it over here. Yeah, so this is neuron software in which you can design your electrical neuron. And OpenVibe is the one which helps you interpret the data and converts it into a machine learning program, which is designed in C++, but also has a perfectly amazing GUI. Yes, any other?
Excuse me, sir.
Yeah.
We are dealing with human brain. So how much
I'm
risk?
sorry. I couldn't hear you. Can you say again, please?
We are dealing with human brain.
Woman brain? Human brain. Right. Okay. Yes.
How much risk is involved in this?
Well, okay. If you're talking about non-invasive BMI, there's actually not much risk. That's the reason you can get these cheaper devices on Amazon or somewhere else. So it's not much risk at all. But of course, when you're talking about an invasive BMI or invasive EEG, there is really sufficient risk. And as you can see, like, these are only conducted in patients where they really have no other hope, and it's still an experimental phase. But if you just want to design a regular BMI with your, like, with the available options that you have, just go ahead, just do it. It's very easy.
Okay, and how the connection between neuron and the electrical signal means takes place inside means anything related to that?
I'm sorry, neurons and electrical signals
Yeah,
with which one?
connections, internal connection between brains, neurons, and the electrical connections.
You are talking about how neurons are connected electrically, or
Yeah,
how the okay. So as I was okay, maybe I just go back to the PowerPoint again. So these are like the first slides over here. So if you would have seen this first slide, so this
yeah,
is how neuron looks,
yeah,
and yeah.
it is okay, sir. But I
Mm-hmm.
want to design a prosthetics. Okay,
I'm sorry, it's just getting broken. Can you just say again,
I
please?
want to design a system which will connect
Mm-hmm.
with brain, with my
Mm-hmm.
neuron. That
Mm-hmm.
system, how it will communicate with my neurons.
Right, I get the point right now. Okay, so you're talking about like a situation like like over here, like when you or a situation over here when you can get the information back into the brain. Yes,
Yeah.
so this is based again on understanding which part of the brain works how. So you have the brain map over here. This is not a pure brain map. This is actually a EEG electrode head map, but it will tell you about which region is involved for which. So this comes from our understanding of how the human brain works. So if you have, so there is the primary motor cortex somewhere over here, and this primary motor cortex is the one that is involved in the motions. And then you have the somatosensory cortex, which is involved. So what it does is that it's still in the experimental phase. That's the reason I'm telling that we do not know exactly pinpoint, but you can send in impulses which mimic the impulses of the neurons which are firing over here, like a potential action potential, which will be able to generate an action potential in a neuron. So you can send a electrical signal over here, and that neurons will fire over here. So with the help of electrodes, you can
Okay.
actually place like as I told over here, I have a recording microwire array, right? So you with these you can also input the signals into the brain. It's vice versa. You can get the signals out, and with these electrodes you can also give so in fact this still happens right now. So people who go with say having epilepsy. So they have intracranial stimulation. So they place an electrode inside the brain and they
Yeah.
stimulate the brain. But then these intracranial stimulations are not directed. So like they do it in a region. But when you have like a very finer electrode, so you can actually go to the specific cortical region of the brain, which you will be designing first by seeing the MRI scans or the EEG scans to understand that there will be subpopulation of neurons that will be responsible for a certain act. And then, like you will be giving firing of the neurons over there by sending in an impulse, which will be generating an action potential of the neurons, and thus get the feedback into the brain or right into the brain. I hope this answered your question.
Yes, sir. Very